In this second video, we're going to look at how DNA replicates, because obviously that's a very important job that DNA has to be able to do in order to pass on its DNA to another cell in mitosis or to another organism in the process of meiosis, where you have the recombination. So what we're looking at here is DNA replication. And for DNA replication, each strand of DNA, which we've already said that each of those strands of DNA, so when I say a strand of DNA, I mean the sugar phosphate backbone with the nitrogenous bases sticking out from it. That describes one strand. And then the other strand is that complementary sugar phosphate backbone with its nucleotides sticking out. So these are complementary strands of DNA. And for DNA replication, what's going to happen is each of these complementary strands is going to serve as a template strand, which is also sometimes called the parent strand. And it's going to be used to, it's going to be the template that allows you to then build a new strand of DNA from it. And so what happens is, first you have to separate the original strand of DNA. So shown here in red, or initially it was red before I started coloring over it, shown here in red is the original parent strand of DNA, and here is where we have started to pull them apart. This is called the replication fork. So they're no longer hydrogen bonded between these complementary nitrogenous bases. They have now been separated. And so what's going to happen now that they've been separated, each of these nitrogenous bases is willing to form a hydrogen bond with its complementary nitrogenous base. But its complementary nitrogenous base has been pulled away because we've separated those strands. And so what happens is free nucleotides, such as this little C here, will come in. It'll get really close to and hydrogen bond with a complementary base on that parent strand. And so the first thing that happens is they form a new hydrogen bond first. So that kind of lines it up. It says, oh, hey, I'm a cytosine and I am a perfect match for you, guanine. We can hydrogen bond just perfectly together. And so that kind of stabilizes the cytosine in this position. And then you can form a covalent bond for that sugar phosphate backbone. So it'll get locked into another nitrogenous base. It'll form a covalent bond at its between the sugar and the phosphate of the nucleotides. So this is an important thing to keep in mind when you're looking at a strand of DNA. The sugar phosphates are all covalent bonds, so that's a very strong bond. The nitrogenous bases are connected to one another by a hydrogen bond, which is a relatively weak bond. Okay, so as we begin to talk about how DNA replicates itself, the first thing that always happens is you have this point where the two strands, so in this picture we're seeing the two complementary strands oriented in that helical fashion, so they're all hydrogen bonded together. The first thing that's going to happen is you need to start to separate the two strands, and this happens at what's called an origin of replication. This is where the two strands break their hydrogen bonds. So they lose their hydrogen bonds and they begin to unwind. This forms what are called the replication forks. So it's kind of this fork in the road where the two strands are now separated from one another. Is called the replication fork. And if you put the origin of replication, if you put this point where you've started to separate the two from one another, kind of right in the middle, instead of at one end, but you put it right in the middle, which is what happens, then you're actually going to be forming two replication forks. One that's splitting the DNA this way, and the other that's splitting the DNA this way. So we've got two replication forks where at the origin of replication. And for eukaryotic organisms, which tend to have a lot of DNA, this won't happen just in one spot. This will happen in multiple spots along the DNA so that we can replicate the whole strand of DNA fairly rapidly. And so that's shown here. There's going to be multiple origins of replication, and they're going to basically move along until they meet with one another, and then you have replicated that DNA along that full length. And so ultimately in this last picture, now we have two identical strands of DNA, each with a new complementary strand of DNA attached to it. So if you kind of follow the colors here, red is showing that original DNA. This is called the parent strand. And then this blue color is showing us those new nucleotides that are being added in. And as those new nucleotides are being added in, you're going to elongate that at the replication fork until you have a whole new blue strand complementarily paired to the original red strand. And so now this sequence of DNA is perfectly identical to this sequence of DNA and to this sequence of DNA. And both of them have two strands. So it's got complementary, and this has got complementary, and this has got complementary. They all have two strands, and all of these are identical. These guys are identical to each other, and they're identical to the original parent strand. Prokaryotes, such as bacteria, tend to have plasmid DNA or circular chromosomes. And so when bacteria replicate, they just have a single origin of replication. So they have one point where you start to separate the two strands, and then you move away from those points. You move down the replication fork until the two meet, and then you have replicated that DNA, and you now have two identical pieces of DNA that are also identical to the original. Okay, so this kind of gives you a big picture. What we're going to do next is we're going to slowly move through and see how this process happens. And in looking at how this process happens, we're going to see these players, these different proteins are each going to be involved. And they each have a role to play in this. So this is kind of our cast of characters that does this job that I was just describing. I'm first going to talk about these first three in the next picture, and then we'll kind of get into these ones, and we'll conclude with this final player, the DNA ligase, in the next video. Okay, so starting, we're going to talk about these first three: DNA helicase, the single-strand binding proteins, and topoisomerase. We're going to see all three of them as we begin to kind of etch out what's really happening in this story. So to kind of orient, we're looking at a strand of DNA. So here's the sugar phosphate backbone, and here's the sugar phosphate backbone of the other strand. And then between these two strands, we have a nitrogenous base sticking out here, such as C, and then the other strand has the other nitrogenous base, such as guanine. And so these guys are paired all down the two strands. And so we have hydrogen bonds between those nitrogenous bases, and then these sugar phosphate backbones are covalently linked phosphodiester bonds. Okay, so we said in order to replicate DNA, the first thing you need to do is break these hydrogen bonds. So we actually need to pull the two strands apart. That means breaking the hydrogen bonds between, for example, a cytosine and a guanine. So we're going to break those hydrogen bonds. And to do that, the protein that does this job is called DNA helicase. What DNA helicase does, you can see it here, it's a protein that kind of fits onto the DNA molecule. It binds to one strand of DNA, and then it moves along that DNA, separating the two strands. So it attaches to, if it attaches to this strand, it moves from the 5' end towards the 3' end. So it moves in a specific direction. But as it does that, it's actually breaking these hydrogen bonds. And it does require DNA to do this job. So DNA helicase is the protein that actually separates the two strands. Now, as soon as you separate these two strands, you have this situation where I'm kind of in the tails of helicase, you've got this free cytosine over here, and then on the complementary strand, I'm not exactly sure where it might be in this picture, but you have the free guanine, and these two guys would love to reform a hydrogen bond. And if we allow them to get close enough to one another, they will absolutely reform that hydrogen bond. So in order to prevent these two strands of DNA from reattaching to one another, reforming those hydrogen bonds, there's going to be a series of single-strand binding proteins. I think in this picture they kind of look like shrimp, but they're not. They're proteins, and they actually grab hold of these free nitrogenous bases, or the exposed nitrogenous bases, and kind of wrap themselves around it, forming these little covers. So nope, there's going to be no reforming hydrogen bonds. We're going to cover those up. Can't see that, can't reform those hydrogen bonds. So that's the purpose of having these single-strand binding proteins. Okay, and then finally, the last player here is DNA topoisomerase. Topoisomerase grabs hold of the entire DNA. So you can kind of see this protein wrapping around the whole DNA. And what it's going to do is it's going to travel just a little bit ahead of the replication fork, which is being made by DNA helicase. As it rips those two strands apart, this right here is that replication fork. Like a fork in the road. This is our replication fork. And what topoisomerase does is it travels just ahead of it, alleviating the coiling that could be caused as you rip these two guys apart. So keep in mind that this coiled DNA, as you separate them, you straighten out this piece, it causes this remaining coils to kind of build up that tension. If you ever played with an old phone cord, back in the day, phones would be attached to their receiver by these coiled plastic wires, which maybe a lot of people now haven't seen them, but they were these coiled plastic wires. And when you would talk on the phone, you would kind of stretch them out. And as you did so, as you stretched out a piece of it, you'd kind of end up with this tangle in the other parts, because as you're straightening part of it, kind of that helical kind of has to go somewhere. And so the purpose of topoisomerase, it didn't do any good for the old phone cords, but in your DNA, topoisomerase kind of smooths this out. It prevents all of that coiling from disrupting the DNA upstream of the replication fork. Okay, so these are our first three players: the DNA helicase, the single-strand binding proteins, and then topoisomerase. This sets us up for the next protein, which is DNA polymerase. And DNA polymerase is arguably one of the most important enzymes in this process. So what DNA polymerase is going to do, kind of is hidden in its name. It's going to make the new polymer. And what I mean by that is, okay, so now we've got this strand of DNA separated. It's got all of these nitrogenous bases kind of sticking out, exposed, ready to form a hydrogen bond with something. And then floating about the cell, you have these free nucleotides. And so these are the incoming nucleotides, and they're technically called triphosphates. We'll talk about why momentarily. So these free nucleotides, what they are is just a sugar with its little phosphate, and then its nitrogenous base, like we saw way back in the very first picture when we were looking at the monomers of a DNA. So the monomer is a nucleotide. So these are those free nucleotides. We kind of affectionately call them nukes. So these are the little nukes. It's got a free nitrogenous base prepared to make a hydrogen bond with the complementary nitrogenous base. And then kind of exposed here on our template strand, or our parent strand, are these nitrogenous bases that are also looking to make a hydrogen bond with something. So what happens is these free nucleotides that are kind of floating around, they kind of float into position and get lined up and maybe kind of stabilize themselves with that weak hydrogen bond. So they're like, okay, cool, this is who I want a hydrogen bond with. And yeah, me too, I want a hydrogen bond here. And so they're kind of lining up based on their hydrogen bonds. And remember, those hydrogen bonds between C and G, there's spots that they're going to make 3. So C and G will preferentially hydrogen bond with one another. And between A and T, there's spots for 2 hydrogen bonds. So they're kind of like puzzle pieces. They're going to find their mate. They're going to find the one that they complementary, where they can form those hydrogen bonds with. So they kind of line up by those hydrogen bonds. And then what DNA polymerase does is it makes these monomers into a polymer by covalently attaching the sugar of one to the phosphate of the next. So that turns these monomers into a polymer. And so you can kind of see in this picture, here's that intact DNA with its 2 strands, and then here's this giant blob of DNA polymerase. It's got itself all wrapped around the DNA. And then here inside here with this little bright yellow color, they're showing this is the site where we have done a covalent linkage. The nucleotide has come in and lined itself up based on the hydrogen bond, and then polymerase comes along and snaps it to that sugar phosphate backbone, forming that covalent bond, making it into a polymer. So it's covalently linking the nucleotides. Now, DNA polymerase moves with staggering speed. And when I say staggering speed, I kind of feel like this is slightly difficult to imagine, because in a bacteria, DNA polymerase can attach 500 nucleotides in 1 second. And so just think for a second about how quick a second is. Let's count it. 1. That's a second. 500 nucleotides could have been snapped into place. I can't even do that many dots for 500 nucleotides in 1 second. Eukaryotes are a little bit slower. Its polymerase can lock in 50 nucleotides per second. And I still feel like that's pretty fast. And if I count to 1, I cannot make 50 dots even on the screen in 1 second. So this whole thing moves very, very quickly. But it does have one rule. It only operates in the 5' to 3' direction. And I think we're going to see why in the next picture. So in this picture, we can see those strands of DNA a little bit better. Let's kind of orient. This is considered the parent strand for this picture, so kind of shown here in red. You can see its sugar phosphate backbone, and there's its nitrogenous bases sticking out from it. Here comes the template, or the new strand, the daughter strand. And so here, for example, is coming a new nucleotide. Here's its sugar, here's its phosphate, and here's its nitrogenous base. The first thing that happens is its nitrogenous base, which in this case is cytosine, is going to kind of line up with the parent's free nucleotide, which in this case is guanine, and they line up together because, as I said, they form these hydrogen bonds with one another. So that kind of stabilizes. Cytosine is like, cool, yeah, I belong here, and it's kind of stabilized because of those hydrogen bonds. But it's not yet linked here. So this is where we need to make the covalent linkage. And if we look at what's happening on this building, this is the strand that we're building, the free end is this 3' end. And then for the incoming nuke, it's got to lead with its 5' end because that's where the phosphate is. And what's going to happen is this phosphate is going to bind to this 3rd carbon. So right here's the carbon 3. So this is the 5th carbon of this free nuke with its phosphate. The 5th carbon has a phosphate. We're going to get a bond between this phosphate and this 3' end. Now, one thing that you've probably seen in this picture then is this incoming nucleotide actually has more than one phosphate. It has 3 phosphates, which might sound a little bit familiar, particularly if you're thinking about ATP, which is called adenine triphosphate, which we spent a lot of time in cellular respiration talking about. Well, it follows that there is also such a thing as cytosine triphosphate and guanine triphosphate and thymine triphosphate. So ATP is not the only one. And what's happening here, just like ATP we talked about previously, having 3 phosphates gives it more energy. It's considered a high energy molecule. Well, each of these guys having 3 phosphates also gives them increased energy. And what do they use this energy for? They use this energy for a endergonic process of forming this bond between the phosphate and the 3' end of that sugar. So we need 3 phosphates, but what happens when these guys bind is we're actually going to break off these 2 phosphates. And when these guys break off, this releases energy. And this energy that's released is used to form this bond between the remaining phosphate and the 3' end. So we need energy to make this covalent linkage. And we get that energy by breaking off 2 phosphates, just like you would have with an ATP molecule. But any of the nucleotides can be 3 phosphates. So they each are bringing in their own energy so that they can make this covalent linkage. Okay, so what's happening is first they hydrogen bond. That kind of stabilizes the nuke, kind of lines it up. Then the DNA polymerase, remember that's that big enzyme that we were kind of tracking. It's this big enzyme that actually facilitates this reaction. The DNA polymerase is going to break the bond for the phosphates, releasing these 2 phosphates and releasing energy in the process. So breaking off these guys, I said releases energy. That's our exergonic reaction. That provides the energy to attach this phosphate to that 3' end. Okay, so this is why when you're growing a strand of DNA or when you're adding nucleotides to the end of a DNA, you can only grow the DNA, or lengthen maybe would be a better word. You can only lengthen the DNA in a 5' to 3' direction, because always those incoming nukes, they have to lead with their 5' end. And then that's always going to leave the 3' end exposed. So that 3' end can then bind with a new 5' end that's leading with its phosphate. So the 5' end is what has these phosphates, which are the high energy. And the 3' end is got the kind of that piece that you need to attach it to. Okay, so this is a really important rule. You can only grow DNA in the 5' to 3' direction. There's one final rule to talk about, and this final rule is DNA polymerase is only able to link nucleotides together if it starts with what's called a primer. And this primer, so the primer has to actually be made first. The primer is actually made out of RNA. So in this picture, in yellow, this is actually an RNA primer. And just to give you a tiny idea to remember RNA is slightly different than DNA because DNA sugar is called deoxyribose. RNA is slightly different because its sugar is just ribose. So it's a difference between deoxyribose nucleic acid and ribonucleic acid. Slight difference. But in order for DNA polymerase to bind to the whole growing DNA, it has to have an RNA piece to attach to. It's because of the structure of the polymerase. It can't bind to 2 strands of DNA. It can only bind to 1 strand of DNA and then an RNA primer on the other end. The molecular, the 3D nature of the molecule is just slightly different, so that this is a rule for DNA polymerase. It has to have what's called a primer in order for it to attach. So what actually happens before DNA polymerase starts getting busy and doing its work is you first have to build this RNA primer. And you do so with, this is one more of our proteins from that list, DNA primase. DNA primase is an enzyme that does this job of linking these RNA nucleotides to their complementary DNA. And this builds a RNA primer. An RNA primer is short. It's typically only 10 to 12 nucleotides, but it's absolutely essential that you have an RNA primer in order for DNA polymerase to kind of bind to and get busy doing its job of lengthening this new piece of DNA. It's got to have that primer to start with. Kind of weird, but very important. So it's a rule that just to keep in mind. So we're working with these two rules. You have to have a primer, so that has to be put on by DNA primase. And then the other rule is we can only add nucleotides in the 5' to 3' direction. This rule here with the 5' to 3' directionality causes us to have what's called a leading strand and a lagging strand. And I'm going to discuss that in the next video.